Chapter 3 will discuss much more detail than the previous two chapters. An overview of types of microscopes will lead into a comparison of prokaryotic and eukaryotic cells. Then we will discuss more in depth what makes eukaryotic cells work so efficiently. Before you read, don't forget to go over the objectives at the beginning of each section in the chapter. The first microscope was very crude and difficult to truly see a specimen, but it has evolved over the past 400 years to produce phenomenal images that help biologists, especially microbiologists. There are two types of microscopes which are common in a biology classroom: the light microscope and the dissecting microscope. The light microscope views very thin samples, usually having been dyed with a stain. These microscopes allow a much higher magnification than a dissecting microscope, which is more beneficial when looking at larger objects such as bugs or a whole leaf. Two features important with a microscope include magnification and resolution. Magnification is how much larger the object appears compared to its actual size, while resolution refers to how clear the object appears. As a comparison to the human eye, a light microscope can show 500 times the clarity the human eye can see. A light microscope, as seen on the left, is used for microscopic specimens which are very small and very thin, such as cells. It can magnify up to 400 times the original size, or if using immersion oil, it can magnify up to 1,000 times. These light microscopes have a stage situated above a light source. A dissecting microscope, as seen on the right, has a light source both above and below the stage and is used for viewing thicker objects. A dissecting microscope can only magnify up to 80 times the object's original size. In comparison to the first light microscopes, which have been around for hundreds of years, the electron microscope is relatively new, having only been developed in the mid-20th century. The microscope uses beams of electrons to create an image. To prepare a specimen for use with an electron microscope, the specimen is killed in the process, therefore it is not possible to view live cells with this type of microscope. And in comparison to the 500 times resolution of the light microscope, an electron microscope can produce an image with a resolution 10,000 times greater than that of the human eye. In the left image, the Salmonella bacteria under the view of a light microscope. In the right image, the electron microscope shows greater detail of the Salmonella bacteria, which are shown in red. These images should give a clear indication of how much greater an image the electron microscope can produce compared to the light microscope. Thanks to the microscope, scientists discovered the cell, which comes from the Latin word meaning small room. In the 1830s, two scientists proposed the cell theory, which still stands today. The cell theory states that 1. Cells are the basic unit of life, 2. All cells originate from other cells, and 3. All living things are composed of one or more cells. There are two types of cells: the simple prokaryotic ones without a nucleus, and eukaryotic, which are more complex and have a nucleus. Remember the pro-no and eu-nu memory trick to keep these straight. For a cell to be considered cellular, it must have all 4 of the following components, each of which we will discuss in more detail. A cell must have a plasma membrane, which can also be called a cell membrane, cytoplasm, DNA, and ribosomes. All prokaryotes are single-celled and include all organisms in the 2 domains, bacteria and archaea, which are the most ancient of all organisms. These cells lack a nucleus and have no internal organs or organelles that have their own membrane, making it understandable how they would be smaller than eukaryotic cells. Of these prokaryotes, true bacteria can have a cell wall outward of the cell membrane like plants. There are 3 structures which help a prokaryotic cell perform necessary functions. The flagellum helps with movement. Fimbria, not shown on this diagram, allow them to attach to surfaces, and pilus, which aid in transferring genetic material. Now that we have discussed prokaryotic cells, but before we jump into eukaryotic cells, look at the diagram and compare the sizes of the cells and cellular components. First, look at the brackets at the bottom to identify what is needed to view each object, either a microscope or the naked eye. Notice to the far left, the atom, the smallest unit of matter, can only be seen with a highly detailed electron microscope. Then at 1 micrometer, that sort of looks like a U and an M, there is bacteria, which we just discussed. It is big enough to be seen with just a regular light microscope, but still too little to be seen with the naked eye. Now look at the animal and plant cells, which are much larger and on the verge of being visible to the naked eye. And finally, the only cells that can be viewed without a microscope are those of an egg. Use this comparison chart to remember that eukaryotic cells, eggs included, are much larger than prokaryotic cells. Although eukaryotic cells evolved from smaller prokaryotic cells and have adapted complex transport systems, there are limitations to future cell size increases. As the surface of a cell increases, it requires more volume to maintain function. Eukaryotic cells have evolved membrane-bound organelles that increase surface area to help maintain life. Watch the linked video for a demonstrated explanation. Complex multicellular organisms like plants and animals are composed of trillions of eukaryotic cells. These cells have a nucleus as well as other little organs known as organelles that can perform specialized functions and are enclosed in their own membranes. Looking at the comparison diagram of a eukaryote and a prokaryote, you can see the complexity differences between the two. Note the simplicity of the prokaryote and a lack of membranes around each internal structure. All cells have a plasma membrane, also known as a cell membrane. This is a layer which helps hold the cell together and is made of 2 layers of phospholipids, a bilayer. If you recall from Chapter 2, a phospholipid bilayer has a hydrophilic head and 2 hydrophobic tails. These 2 layers form a barrier that is selective or picky in what it allows to pass through. Between the cell's plasma membrane and the nuclear region is an area known as the cytoplasm. This area houses all the organelles which we will discuss in this chapter. The gel-like substance in the cytoplasm area is called the cytosol. So, the cytoplasma area is full of cytosol fluid. Also in the cytoplasm region, there is an extensive network of protein fibers that help facilitate the work performed inside the cell. This network of fibers comprise the cytoskeleton. It has 3 types of fibers which serve different functions, including providing shape and transport of cellular components. What do you think cyto means? All 3 of these words, cytoplasm, cytosol, and cytoskeleton, are found in the cell. Cyto refers to cell. These 3 protein fibers also serve in cellular movement and division. The flagella and cilia are both hair-like structures. If you notice from the image, flagellum are long and help move the cell around. Cilia are short fibers that are found all along the surface of some cells and can assist in moving a cell along the surface of an object. Centrioles are fibers which are important in cell division, and a pair of centrioles make up something called a centrosome. These 2 we will discuss more in a future chapter when we talk about cell division. The endomembrane system includes the organelles and membranes which work to process lipids and proteins in the cell. This system includes the nuclear envelope, lysosomes, vesicles, endoplasmic reticulum, and the Golgi apparatus, all of which work together to accomplish life functions in the cell. Inside the nuclear membrane is the most important organelle, the nucleus. It is so important because it houses the cell's DNA. DNA is found as chromatin, which then forms chromosomes. We will discuss this concept much more in Chapter 9, so do not worry about it if it's fuzzy right now. In the nucleus, DNA is rewritten or transcribed into a smaller molecule known as RNA, which is used to build proteins that help the cell do work. Inside the nucleus is an area known as the nucleolus. This area helps synthesize or create proteins with the use of ribosomes before sending it out of the nucleus. This transcription is important as DNA cannot fit through the nuclear pores, but RNA can, and therefore the instructions coded on DNA needed for the cell to operate are able to get out to other parts of the cell. The endoplasmic reticulum, or ER for short, is a folded network of membranes which are instrumental in modifying proteins made by RNA, synthesizing lipids, carbohydrates, and hormones, as well as forming enzymes which help to detox poison and metabolize alcohol. There are two portions of the ER. The rough ER, which is so called because it is covered in ribosomes and therefore is responsible for protein synthesis. In the hollow interior, proteins are folded, or molecules are added if needed, and then secretory proteins help secrete the proteins out of the cell. The rough ER also makes phospholipids, which are necessary for the cell membrane's bilayer. The ER lacking ribosomes on the surface is known as smooth ER. This area is responsible for synthesizing carbohydrates, lipids, and steroid hormones. It is also the site for creating enzymes which help detox the body of poisons and/or alcohol. The Golgi apparatus, or Golgi body, is an area of folded membranes where proteins, carbohydrates, and lipids travel to be sorted and routed to a final destination, both in and out of the cell. Lysosomes are only found in animal cells and are referred to as the garbage disposal as they break down worn-out cells, bacteria, and other cellular structures. In the image, an immune cell known as a macrophage detects a bacterium cell. The macrophage engulfs the bacterium, and then a lysosome attaches and breaks the bacterium down, and it will then be expelled as waste, both from the cell and the organism. Vesicles are useful in transporting cellular components, while vacuoles are used for storage. Ribosomes are essential in the production of proteins, which then become the instructions for the cell. These are found in all types of cells, since all cells synthesize proteins. They are made up of two parts, a small subunit and a large subunit. The mitochondrion is a unique organelle in eukaryotic cells. It has a double membrane of phospholipids. It also has its own DNA and ribosomes and is known as the powerhouse of the cell, as it is the location of cellular respiration. Cellular respiration is the process where glucose is converted into usable energy known as adenosine triphosphate, or ATP. Inside the mitochondrion are two important areas that each have a specific function for cellular respiration. The cristae are the numerous folds visible in the image, and the matrix is the area surrounded by these folds. We will discuss cellular respiration in the next chapter. Peroxisomes break down fatty acids, which are the building blocks of lipids, and amino acids, which are the building blocks of proteins. They also aid in detoxing the body from poisons and alcohol consumption. When they break down the toxin, hydrogen peroxide, which causes damage to cellular components, is formed. However, the peroxisome has enzymes that can then break the hydrogen peroxide down into water and oxygen. Although both animal and plant cells are eukaryotic, meaning they both have a nucleus, there are several organelles which make them distinct from each other. Of the organelles which we have just discussed, animal cells do have centrioles, centrosomes, and lysosomes, while plant cells lack these structures. And plant cells have a few structures which animal cells lack. Plant cells have a cell wall, chloroplasts, and plasmodesmata. They also have one large vacuole, which is different from animal cells, in that animal cells have numerous smaller vacuoles for storage. Watch the video for an animated comparison of animal cells versus plant cells. Plant cells, protists like amoebas and algae, and fungi have a cell wall. This wall is on the outside of the cell's membrane and is composed of cellulose. The cell wall serves to protect the cell in addition to providing structure and shape. Any organism that can capture the sun's energy and convert it to glucose is able to perform photosynthesis. This process occurs in chloroplasts, which are the green structures in the image. Similar to mitochondria, chloroplasts also have a double membrane as well as their own DNA and ribosomes. In the inner membrane of chloroplasts, there are stacks of sacs called thylakoids. Each stack is a granum, or grana for plural, and the fluid between the stacks is called stroma. These structures play important roles in photosynthesis, the conversion of solar energy to glucose. We will discuss this in more detail in Chapter 5. As previously mentioned, plants have a cell wall which provides structure and shape, but they also rely on a single large vacuole to help maintain shape of the leaf or plant. This organelle is responsible for regulating water as well as storing it, which gives the cell a plump shape. This plumpness pressure happens due to turgor pressure. As a reminder, turgor pressure is the pressure inside the cell which pushes against the cell wall, giving the cell shape. When water is limited, the vacuole will shrink and the plant will appear wilted. In both animal and plant cells, there is intercellular transmission of material in addition to communication across the cells. Animals have an area known as the extracellular matrix, which is outside of the cell. They also utilize tight junctions, desmosomes, and gap junctions to aid in these cross-cellular functions. Plants only have plasmodesmata to help with transport of materials and communication. The area outside of the cell is a maze of glycoproteins and collagen known as the extracellular matrix, or ECM. This area helps to hold cells together and form tissues, as well as facilitate communication between cells. For cells to transport materials between nearby cells, there needs to be some type of connection or junction. In animal cells, there are three types of these junctions. Tight junctions that hold cells tightly together to prevent leakage between cells, desmosomes that serve as little points of glue or welds in tissue that needs to move and stretch like a muscle, and gap junctions that are channels between cells that allow movement of cellular components between adjacent cells as well as allow communication between cells. In plants, transport and communication is accomplished through plasmodesmata, which, like gap junctions in animal cells, are little channels that connect two adjacent plant cells. These channels must pierce the cell wall to accomplish the task. In this diagram, notice in image A that the break in the cell wall between the two cells is allowing passage of the red arrows. This is plasmodesmata and is only found in plants which have a cell wall. In images B, C, and D, these are all animal cells. In B, the tight junctions hold the two cells tightly together. In C, desmosomes are connections that allow organs and tissues to stretch, and D are gap junctions that are also channels between cells similar to the plasmodesmata in plant cells that allow passage of substances. When you hear the word mosaic, an image similar to this one hopefully comes to mind. When you hear the word fluid, you should think of the ability of substances to flow. In a cell membrane, the many components work together to create a mosaic that allows for substances to move across the membrane. Just like the artistic version of a mosaic, the plasma membrane is riddled with an asymmetrical positioning of cellular components. Remember, a cell membrane is made of two layers of phospholipids, which have hydrophilic heads to the exterior and two hydrophobic tails in the center. These two layers have proteins, carbohydrates, glycolipids, glycoproteins, and in animal cells, cholesterol, positioned around the membrane, forming a mosaic. The orderly arrangement of phospholipid molecules make the cell membrane a liquid crystal, which allows molecules to move rapidly. Lipid bilayers are flexible, self-sealing, and confuse with other membranes. These properties together make the bilayer membrane mosaic and fluid. Watch the linked video for an animated visual. The proteins intertwined through a membrane serve several important functions for the cell. These proteins or protein channels facilitate transportation of materials. They can serve as enzymes that alter the reaction times in the cell, and they can help link cells together. Cell membranes are designed to contain essential material inside the cell and prevent unwanted material from getting in the cell. However, there are some molecules that need to move in and/or out for the cell to survive. The membrane allows this to happen by being selectively permeable, meaning it is picky about what can enter and what can exit the cell. The membrane helps control these materials based on the composition of the molecule trying to pass in or out, thanks to its hydrophilic and hydrophobic regions. It recognizes polar and nonpolar molecules, which, remember, is how evenly distributed electrons are in a molecule, and therefore the membrane can help control the amount of molecules on either side of the membrane through passive or active transport. Passive transport does not require energy and includes diffusion and osmosis, while active transport pumps materials across the membrane and therefore does require energy. Endocytosis and exocytosis, which we will discuss in the next section of this chapter, is considered active transport as well. Passive transport does not require any energy, but instead relies on a concentration gradient difference to work. This means that if concentrations of materials are different on the two sides of the membrane, movement will occur from the higher concentration to a lower concentration. Not only does diffusion work across the cell membrane, but also inside the cell and in the air around us as well. If you spray something in one part of the room, it will eventually diffuse across the room into areas that are lower concentration until the spray molecules to the air molecules are equal. How fast diffusion occurs is affected by how different the concentration is on either side. The greater the difference in concentration, the the faster the molecules will move. Smaller molecules diffuse faster than larger ones, and as density goes up, the rate of diffusion goes down. Facilitated diffusion works the same way, but is facilitated or helped by proteins in the membrane. Notice in the diagram that when there is a greater concentration on one side, as is the case to the far left, the molecules will move across the membrane until the number of molecules is equal on both sides of the membrane, as is to the far right. Diffusion and osmosis are similar in that they both happen without the use of energy expenditure, but while diffusion spreads solutes, which is the substance that is dissolved, osmosis spreads out the solvent across a semi-permeable membrane. The solvent is the liquid portion. Notice in the diagram, in the left picture, the beaker has equal levels of solvent or liquid, but unequal levels of solute or particles on either side of the membrane. As a result of osmosis, the level of solvent has changed so that the concentration of particles in the liquid is now even on each side of the membrane. The liquid moves in osmosis. How much solute is in a solution is known as tonicity, and how much dissolved is osmolarity. There are three terms to describe the differences in concentration of solutes: hypotonic, hypertonic, and isotonic. It is easier to remember these three terms if you remember what their prefixes mean. Hypo means lower, hyper means higher, and iso means equal. A lot of people are familiar with diabetic terms such as hyperglycemic or hypoglycemic. If you're hyperglycemic, you have high blood sugar. If you're hypoglycemic, you have low blood sugar. If you think about kids with a high level of energy, you might call them hyper or hyperactive. Knowing these prefixes will make this idea simpler to understand because they can seem backwards when thinking about it from the cell's perspective. When discussing tonicity, we are talking about the concentration of solutes in the environment outside of the cell. So if you think about this with salt as the solute in these cells, it is a little bit more understandable. It is very important to remember that the tonic portion is the extracellular fluid. In the diagram, the first cell is in a hypertonic solution. This means there is a greater amount of solute in the area outside of the cell. If the solute outside of the cell happened to be salt, what would you expect to happen to the water content in the cell? If you look at the cell, you can see the water is pulled out of the cell in this case. A higher concentration of a solute, like salt, would pull water via osmosis into the extracellular space outside of the cell. When this happens, the cell loses water and shrinks. So hypertonic causes water loss in the cell because it is needed to regulate a high level of solute like salt outside of the cell. Notice with isotonic, which meant equal concentrations, the arrows are going back and forth because the concentration of solutes is equal both inside and outside of the cell, so the size and shape of the cell remains constant. With hypotonic solutions, the area outside of the cell has a lower concentrations of solutes, like salt, and therefore the inside of the cell has the higher concentration of solutes. In this case, the excess solute inside the cell will pull water from the extracellular space. This can cause swelling of the cell to the point it could burst. So hypotonic could cause a cell to explode. Any of these changes in tonicity changes the turgor pressure of the cell. Go back to slide 32 if you want to see that. While passive transport occurs without using energy and moves substances from a high concentration to a low concentration, active transport is the opposite. It pushes substances from a low concentration to a high concentration, which would require energy. The energy used is adenosine triphosphate, or ATP. The cytoplasm of the cell has a negative charge, while the extracellular fluid outside of the cell has a positive charge. This creates an electrical charge difference known as an electrochemical gradient. Therefore, when positive potassium ions, represented by the purple K+, need to move from the negative interior of the cell to the positive exterior of the cell, it is going against the concentration gradient and will require energy to move it, if you notice the black arrow going through the protein channel. At the same time, potassium ions are being pumped across. Sodium ions, which are represented by the blue Na+, are being pumped across to the negative side. This cross-pumping is known as the sodium-potassium pump, which is critical for cell vitality. There are times when a cell needs to engulf or expel large particles or even parts of a cell. This is accomplished with a type of active transport known as endo or exocytosis. As the names imply, endocytosis involves engulfing substances, while exocytosis is expelling substances out of the cell. Cells have three ways they can engulf large particles: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is when large particles are engulfed or eaten. Pinocytosis works the same way with smaller solutes, and receptor-mediated endocytosis is specific to certain substances that must bind to a particular receptor to be engulfed by the cell. The opposite of endocytosis is exocytosis. This is when the cell needs to expel a large particle or particles. The cell encapsulates the unwanted particle into a vesicle and it is sent out into the extracellular space. Watch the linked video to see these transport mechanisms in action. And finally, don't forget to go through the review questions at the end of the chapter.